talingim oh you tarawra wal balula lembalti kare
Oi, malum tu tau como sabe la radio station el grupo se ve del gut makliti al togo betsel aradro res ala agora bandera se atian tusten dustei me di dulutro amo te plilo se ti agora va special tirgal gesto lo ngedre al tu tau agora war que debate mil me di suadel mo aisa bebir a trentin la urul do mo cortial eutre la klingin la del belau le te mombra la kluguk mong aun mo ngite y agora togo betsel seu ma era do le mol me bagu mulo do sia sus mai bedul maraklai bilum e biraklai arugul rubugul mar wasil belau era do sia togu betel sus mora president vice president ar gabinete ti eringi marugul senators ma al delegates marugul tigel mutringa gal madala aurora amte vidra belau como am sulanga togu betel motriel no sil guktuni do reso tia chorgi al tutau alo ai seti il sergi al tutawa seldari asing understanding biodiversity in Palau's ocean e ogore updates on IDNA kada ora sadel mo ninga wil clouding ergi tial el debel belau ma osi sil time sel tora IDNA IDNA tora tose environmental DNA selbol tokor belang wasay ngod gora wado su baigal as asmel malu ngora taptibil igal ngi le mo mudira say ngara mangaral ngi lakker tial el sela e wotelela klingi la adabed mangaria terul adal motatal ro tial adraga ikrel belau ma malt maru drasa ngal ngal gedra mre ngal ngatia ngadir lo lo bentrit metirga ya gora mo o wai sergi da igel ting tagi dilure otherwise ngari agora sel announcement mongwase sel got announcement ase sel mom lum email anniversary center amorina dustel January 15 rel arrak amina next year coming ra next year 2026 sel datial anniversary gida agora seng lum email ra ogorwila adbet lultuira adra blue ma omsu ma blakarding say agora wal daital modime was 25 years of ocean stewardship rooted in community grounded in science and empowered by commitment say agora wal adat tagi del 25th anniversary was atigeta adikmedu tmostat announce mo trule selogerul el announcement ase ngaringi ate al rauror el gere la tal researcher ngamo orol el gere tegi gagi del dosa de la DNA project el ngar engel sel research department trasentere e wase gel gora morel ngagi del ada maldimo motmok la tora omsub research e direk gora wal manage bali ngora sel day to day operation egel tingira aurora bekel sils tial tora IDNA ma dire tora gel dasen ocean acidification egel kel milada ngaria lab ngagi dal mo aurora ma motmok la gay tora research o sil time ngora wal make sure ala bakal tmok gire la gay tora DNA ma gel tora gal milada ocean acidification ya gomagailom so del tial auror ambal simil dimo visitra a website trablay e ogore tial auror amo musimra e imrgom singila tial melbuil October close October 5th sel tara announcement asay temo tukulmu telmo mru seguid la intercession program ma Ocean Explorers program la September 29 el mo October 1st tia aditaba el program el giri a Royal School mong Monsamil sigum some ako makalom so den simil direk mo mesa sel. En simil tira Facebook page malu ka email sel outreach department apply PICRC. So say outreach.picrc@gmail.com. Agagi ko ram le sub del mosme de lo kuko ay silmel gid altutaw. En shagal mo tial lang ko le mosyo gat gal el guest gid. Ngari angele got ngel gal sal mo mesima bita tial kisiera Collin Closek. Staff scientist sel Stanford Center for Ocean Solution. El ngara Stanford University ra Blura California. Ya tara guest gid altutaw at Jeanine Brantschen nga early career fellow at Stanford Center for Ocean Solution. Dire tangara ko si sil alat el dire na sige del Stanford University ma of course amring aba modeling ngalge del lamutol gi bem dumutiang indire kal malwi lang udra amring leng mungat mok lagal tokora kora arwa MPA masurbe dire kal mo involve gai tokora arwa akela ngawale kora almol amina ngawal mong malwi lang udel sel do mes ngin lo lo mentri gai el hagasel ngara krebel almei leng tatmakaluro o sisita metedire wal lolisi malu betemang lor sel clothing tirgira ga gidro lor malu ngina nudra ga gidro partnership. I would rather have you guys talk about your center, what sort of things are you doing. So let's start with you and then after we'll ask Jeanine.
Okay, great. Yeah, so the Stanford Center for Ocean Solutions, we work on a variety of different challenges within the ocean. But what we center around is conducting research and also innovation of different technologies and ways to basically be able to have impact on the health of the ocean. And over the last couple of years, over 8 years now, COS, the Center for Ocean Solutions, has been working with PICRIC on a variety of different projects. And what we'll be talking about today is the Palau eDNA project that we've been working on.
Okay, Collin, number ko na sa Ocean Science Solution. Al-adal mutmok lagi lurior, temutmok omsu maurior, ilngia wabul sa klitial dabe. Ya osi time ang tedire ko ora omes raseng to batte de tora omsu research e dire ko ora Michael ora esel teletara aurore to sa innovation in science ora esel teletara omsu alo spell tral sa bakra tel daw engage del malmol wasay ya did you say you started working in Palau in 2018
Yes.
Yeah, era 2018 التاجيد الالابتين اقول له مسرسي قام يدوروا المصير بلالي ضرقوا المدينه سيجيد 2018 يغدوا المول امسو ديال المسبلا اوتل الاقلين لا بالبلاو بس يجي غدو سنتر وكل غان الكترومتري مثل مو الغرا اورلو بنجدل يا جيد الغرا ريسيرش يا جيد الغرا كيكارا اوريري رجل بودو صادر لا توتاو thanks kan Jeanine you do mind to tell us I notice you're like the introduction it says early career fellow. Would you mind tell us a bit about that so people can know?
So I mainly collaborate with Collin at the Center of Ocean Solution, and I joined Stanford in February this year. And my background is really more in environmental DNA, so I used the method before in different ecosystems. And then we also use it in our local ecosystem, so in California. I mainly work on a project with Collin that does a similar approach that you do here at PICRIC, but yeah, looking at coastal ecosystems in California and also like deeper waters there. So it's the method itself is very similar, and yeah, my background is more in biodiversity monitoring with eDNA.
Hindi kita sinig lasing aurere lagu ngomsin del Semedula sing ora klol basen traurel angara igai gidel da sing idi ene gal gora as semel ma tap tabil ma kaskele la arma ba enga me ulmu se ongerul bul lorot tagidel aladel da sing oceans Center for Ocean Science ulmu se ongerul bul ya umsindulu de la ngara ngora coastal area California indigal lurere la dire malo se dios ngegi lo sing medel dios sisiu ni maigal dorur belau dire gal lurera coastal egal gora kabo gora kula blu dire gal mora atmol al dago malungar el gamar tilteru el me gora guk mo ay segel blada kat moklo el gire sera al gotlo mulo lo sergi de tel e hotel la da akling la da ba da belau el melmutre la mo very interesting. Thank you both for coming, especially you know the years we've gone through. For example, from 2018, we've been working together, and so we're quite excited. Excited. I was going to say, I guess, but that doesn't sound good. We're very excited to see since 2018 what have we done so far. What can it tells us about the management of PNMS and what can we do to improve or take it to the next step. So thank you both for coming and joining us this morning.
Thanks for having us.
Diaksal mal mo introduce ramon lang kadimdiling ngalaget ma baydo mo mang wala di swadro gala say. Can you tell us since you came in 2018, can you tell us a little bit about our when we first connect and start collaborating on this project?
Great.
So like the background,
Sure. Yeah. So we had a working group that worked with various experts, both in Palau and within the United States and a few other countries that were working towards understanding of what the PNMS would be able to achieve and what the knowledge is that currently was known about the open, the EEZ, the space of the marine waters, and what was still knowledge gaps. And so from that, we identified that there was still a big question about the biodiversity that was in that space. And while there's many methods that could go to examine those types of questions, we identified that eDNA was one method that would be fairly easy to collect and be able to get a lot of information from the data that we collected. And so once the PNMS began, then we wanted to be able to start collecting samples. But that was also at the start of COVID and lockdown. And so ultimately we were able to get some funding to be able to support the work. And then we started collecting. Once the borders opened, we started our first collection in August of 2021.
Okay. Tegi del ladra Collin, Closek Center for Ocean Solution. Sera 2018, radora alaigal tokora si alia 2020, engom musim tel del bela almoda ka umidringi, yegida multau sel ula tel la sing eotre la kring del bela, sel PNMS, Palau National Marine Sanctuary. Segida al musim 2020, engagida Collin ngatiang marwa tel center gid, amlogor omes asay. Mabolong sim ngrai gel gora amo ole la bas el gire la udor tegi del otro la kling la del belau mesela bolo segi denga dengal golen lo ay sasi mboto gar haka sel ngaratel al adaktir magukti bebira kora aladra ablulat kora ablul dage melas mlon sergi del gire tegi tora a PNMS matulmu ora kora al dadu mesela lo mural dadu tora mlo mes lo asay magarang lesel mlegora onti dasay kame musuk mamel masatial asay ngarama ngaral ngigil malubeng ngaral kagarus sebenarnya laklang rakir tial kita mendengar ada bela kita mal dimle insur ko ora umsin dil dengar al sela agar gere dimle al salma al mul mendengar maklaw ngir gidra aikrela amin aikrela al mul Saya ko ramle wala aldo du mo mo lagagi dulu masala lo mo la etegi la ladra wa akalin el Center for Ocean Solution amlo statro mungkin lo ko ram mo crowd ma tara ko ram sorry so da long ringi awasi ngara mangara kera tel debet ngegi da golin wasi mboto ka cross el tretal se los pel mo mira se ngral arma ngral akket tel da dito ko ram lo mti la se tegi dito ko ra EDNA tia la otro gora betengela aurol se edlo malubeng rail se edlo gimi mo mdira sengara maral arma masigi de temlong tik tia gidlo aseng tia boral do gimi kadmo o galekta DNA mo kadmo mdira sengaral ga rus betengela aklanga rava bang al seltial e otlela masigi dralbo engaji del mulgola sei mungora omu lauror enya monia covid mengwal gora trobaro guileng lo de arwa omral mo de sin ngara bluri de me mo de ase de logro mo matral omral engegi de lo sei masel gotra election amle hapenera a sera 2021 se volver a covid mlut el asmele legal bulu la sei mla mo subul doral en digital rakta agora amral mo mitut mla mo se de lo mi bais e tira li lut el mem sal mei etri git muklisel gotel EDNA election sera ragla at 2021 and so when you after the COVID 2021 that's when you start you did the first collection and you've been try to collect sample ever since so you did 2021 start to collect a sample but I believe you didn't necessarily travel every year 2021 22 to collect the samples. I believe you kind of train some of the
So
can you tell us a bit about that?
So a large part of this project is also being able to develop the program as a sustainable program in the long run. So from the very beginning, actually, when we were still in lockdown, we did trainings virtually. So over Zoom, I had sent the whole setup to the team, and basically we went through the steps of being able to collect a sample and being able to filter the sample, and we did that all over Zoom in July. And then once we got cleared to be able to fly, then we came to start collecting the first set of samples. But of course, there's been a lot of preparation and planning up to that point, also with marine law, to be able to go out and collect the samples.
Right.
And so, ever since then and continuing, we have trainings and are building the ability for Pickerick to be able to continue doing this work and be able to use the data to really be able to inform decision making in the future.
I see. Masigi dral masimr ngomul al dadu mo katwa di mo pergi dengan masimra gal bulun mo dak omral mongowai sakali no sei masera bolwai segit yagi kora di miru ragal tose online training katwa di gara kampire dadu kora a tren no sei mo magara tetela urur magara bodo galek taigang mo mok lagi gidil kora training engegi di direklo ai sala sei masera 2021 lagi mai yang direkan mal mereka asulel tial dedung patrol boat tial marine law officer gidra ngar kamalal bay ma kamalal ngal mereka asulil le kadro gul madengek mo ay galuro ra kawal malungila tangara mokrol bilas gidra gul madengela sei tial center gidra da akrol bil sengel se elor tara mo malaga gidra sampol mo gagi direkan mal gora mo make sure ng dulisel plesel marine law sei ada mal mereka asulil se gidra gima 2021 yung mula sa mga malwagi mo bantri nga adilid el motor de mala sampol ma siya nga rega gitu ka sampol ng dola ba yun at time
Wala de so modo alise selo lo ringi ra earlier lo aseng ang dera ngil dong urigid el evel lo spe el mrulat subel ang wa la deep sea research sindel dong ura wa deep sea research obra de otqadospe akren ospe a research vessel maklol research vessel المرول ستجد الإذي إن إيه والغور عملة بيوترول اللي قد مسوب بالجريلا هذا بالبلا ما مدنيلك مونغرا السيل اللي الأصل تقول إذي إن إيه قد ذي أسبقرا نسكين بارلز أفل دي غرا يقال ارتريجي 29 footer Kada di ospa ikagid ni skin barrel deploy ringi yakalek taral meme filter. Edera kalso kalmo emphasize el motel project regida awan regel tara got el project el ngaringi a time series ringi. So el agid akalek tra bekal May ma around May ma November bekal raksin sa the start el الغالك تاو ulan sa edel mula sa kada monitor biodiversity la kada marega gidelo si siwil site galek trege gidel al tamra al sela beqal ra
Oh, that's a very good point. Kakabaram sila kemring. Agagita agel kora wal kora telang tingil. Agagita lulor. Blalo ay sabi niya malungil arungo dile mereng atlo trigagita talek taigi mo. Again, akmutra kwa di mos misi isel wasi maul mong kadamlang ititay del teletail idna tagi del blalo de amring. Mual beot trigida adi ngara ertel motob wispada be malme omi emum de bongara maral ngi lakke selu trigagidel. Lengga seda klol bureng rigidel ola bagel ora underwater sab ngala lola bulsa.
الأسل دبر offshore kada ngara air tramarin lomel misolar atramarin lol blol igid makados pera air titil mokalek tra offshore hindi sel ngara near shoring sa edel di ngara igal migarel air.
Why say the earth rigidel? Why say? So through this, the work you're doing, what are some of the, I wouldn't say sort of like a shortfall or obstacles, some of the things that you face that, or problems? Is there any problems since we?
So, I mean, I would say in general, it's pretty challenging to go and collect these samples. It's one thing to be on the shore and be able to go swimming in calmer waters, but there have definitely been times where we go to collect and it's quite intense weather, storms, etc. But also we go collect very far out, and so it's both really an amazing experience to be able to see how vast the space is, but also recognizing that when we are out, we have to bring everything with us. There's no coming back for anything. And so it just requires a lot of planning and preparation.
Right.
And ultimately, I think it has really made the team congeal and work really well together because we're all dependent on each other to be able to make those trips successful. So yes, it's challenging, but I think we also have been able to figure out a way that works well for us, and we keep optimizing the methods that we do so that we can do things as quickly and as easily as possible with still being able to get the data that we ultimately need to be able to do the monitoring.
Ngay sing mo ko kolin na siya ko ra di mo mrang le gay to ra offshore kada malmor oro dra blue sel mo ngila ba bendre kal malati to koling ertom toa te kamalas mis padao balu ba oi ngora wala ti to indi wasay ngowe gagi duluror etiditil galek tao sisilta betiditil lo mas asay elang ngil do korang mo botri da maigang aidal sampol ngil do korang tirga ko ra wadil til lorore msamok la galurori mo sel gotul betengele ending mo ko lasing sel de top de mo trawan bola me sablu ko ngora mal mo swasel asay tial daba moral Chloe, dia kami sa bulu dia mesel di tabel meleliwe ko sel kwa wal vastness tial ngora mo ko seguido se sel bulu ngora mal mum dia seng makro tial dabe agil ngora wal rangle lauror lorur triga gid ladamo ngora lungi la rut sel da boriate medel me edo mesa gal ngora risalte lengi gagi di risalta subdi la sei ngaria adu no sel mai gal me un mo kadam tingi se gida mal sadar ngora emphasizing sing bring lindit temral lo babla reng mangal mok mo ulen sel matingi gal. And now when we come to like a DNA, can we talk a bit more about understanding a DNA, a bit more detail on how can DNA be used for understanding the biodiversity, for example, or the marine environment?
Okay. So we will go into a bit more technical detail of how the method works. Yes. So DNA in general is the genetic information, and it's basic building blocks that all life on Earth share. So we are built from DNA, but then also bacteria are, fish or marine mammals. So we all share these basic building blocks. However, the genetic information is different between different species. So you wouldn't necessarily have the same DNA as a fish. You can think of it a bit as an alphabet. So you have the letters and they are the building blocks and they're the same. But then depending on how you put them together, you get different words. And it's the same for DNA and different species.
Jeanine nakukul kasi kerto ko na DNA kasi generic building block Saya kasi korawal sa elabatinget. Kakarusa sa elabatinget. Ngagi del mulwa sa kador o mesingil wasing korawa egal alphabet Botokol letters hindi mal diyo kasi angara mangaral letter abol modak emget ngagi del armo. Makuk ngara mangaral letter abol dak emget ngagi del armo. Manggagi del mulko lasi adi ini angari ra roguil ngari ra arma del lom magidarat. Hindi nakal necessary lasi just you see DNA gideling. Saya gitu dulu dia sih, enggak harus DNA amotmak, eguk bongi gila. Enggak harus DNA amotmak Saya kata italom so dela DNA. Lalu dia Jeanine lo sing, it's a building block. Ngiga gitu motmak endub memberal buklu. Mulas kosmetis unique species
الآن قرأت الموضوع اللي say how do we go from water collection المرة DNA processing so الآية تلتلة لاقي مغلق تايقان قدتو ذا المرة ولا غلق water sample إمي مرغن يسلتي so يقال الأرم ويجي ذا الأذى الدرق الشيطة سكينر تير ما ويسي يجي ذا الparticles ang aring iya DNA details at the extract me yak mula si ong kagi ang arang alarm. Yagi ذا DNA detail at the tigal al sel yagi ذا water sample serigid mo ulo kadang malme filter emrong yam extract amos at the meding la sing ngara mangaral alarm mangara al sel at the belbela.
Yeah, as you guys were talking about this DNA stuff and how we just basically collect water sample here, that means some scientists way before you guys has been doing this. Is that right?
That's right.
So now we simply just go out there, scoop the water, come in and look, because there's like a big, how we call that? Like big database of different, is that how this thing works?
Yeah. So, this type of work has been conducted for over 60 years. The majority of the methods were taken from understanding microbes in the ocean. So small bacteria or other microbes, because we can't see those microbes. And so we needed to be able to come up with a way to be able to identify them. And in time, we also realized the advantages of being able to collect DNA of larger organisms as well. And so the whole way that we ultimately identify what the DNA is originating from is from these databases that you're referring to. And so those are usually different voucher species that are submitted to the database. And then when we compare what the alphabet, the building blocks look like compared to the database, then we can match them up to what the organisms are. And so what that means is that we can identify many organisms using the database. What it also means is that there's still a lot of work to do to add additional organisms so that we can continue improving what identification.
Building on the identification. Wow, it's a big, it's a lot of work.
Yeah.
It's a lot of work. Yeah.
It is.
What are some of the advantages of using DNA? I guess one advantage is, again, we don't need to go through this big hustle of like moving a big crane to go over there in an underwater camera and to see, okay, so that's whale, that's what. And again, when it comes to microbes, there's no way you can just do underwater sub or something and identify them. I guess that's one advantage. Are there any other advantages of using DNA?
So I think what you touch upon is we call it, it's quite low tech. So you really just need to collect a bottle of water.
Yeah.
And you can do that across many ecosystems as well. So if you're interested in mangroves, for example, you don't need to use another method than if you want to go offshore, it's still collection of water. So for that, it's an easy method to standardize the sampling effort across all the different areas or zones in the National Marine Sanctuary. And also you can look at different organisms. So for now, the project has been focusing on vertebrates mainly, but there is the possibility to go back if you're, for example, more interested into algae or more bacteria. You can go back to those samples and look at the DNA of that as well. So that's another advantage.
Mralungit kertongora DNA leng ngora beta urel lengar le da faldi mo ma aware sample malmeng e gore o ngariate ale atleta lo sperie mot mautel mi mon gel gora bol model blalbi asil malme ngariate tele dia bo sadrin dia gudeng tira mom sadet tetesi anyway ya uri la da malmong omes emo modengel asi ngang gora asas bela ngaral ar mo asas bela ngaral mo mo dia seng ngaral engge da tenin mo la sei palbet tengeringi adia galwa over omwal over adru la kaburs over agarger over ekra almol mal de sisil medot pamal dingo war sampol me o si da bal klungiolel tiala DNA. Yeah, it's really I think it's really convenient and easy method. I mean, as long as there's a water sample,
Exactly.
you can figure out what's in there.
It requires that you go out and are able to access those environments. So one of the challenges that we've had until recently is being able to get those samples from more than just the surface. And so Meringue was speaking to, being able to go deeper and being able to collect samples from deeper in the water column.
Right.
Because while the waters that we see from land are fairly shallow, it gets really deep. And so we're literally just skimming the surface when we collect samples just from the top. But it is overall conceptually very easy. And then once we get that data, then it's trying to do the hard work of trying to figure out translating that data and also trying to be able to understand what the trends are that we're seeing within the biodiversity.
Right. Yeah, I understand that, scooping water sample from the shallow water and surface is easy. But you're right. How can we try to extract the water sample from the deep? And so...
But then I would also like to add on that we have had opportunities to collect water samples from the deep. Ay gal tabulol del el may narilus mo okeano sing mlaringier atadel el moregal del langaringi adong urtir el sib el marat mol so el asera el nea natilus yatem nemru la ROV survey remotely operated vehicle attached to the ROV gagid el niskin barrels misal temela atel ROV mamateng al yagid direk al asker tir metagalek ta water sample mangaringi water sample rigid el that mol seragim survey gal si mount ra al sala PNMS yagid direk al mle collect ta water sample up to 3,000 meters deep metigid el temel okeanos metidirek al mle galek ta water sample los per sige del rosette tir dongor tir regal si mount.
Wow.
Yeah.
Again, thanks to all this collaboration and partnership from outside, I get to blow the lo comring la seng metial belal membro la jale ko mapping. Comring per agel ko la ship to share interaction with the school. Ayegi ding il dial. Ayemle me mrura chizu. Mle me mlu ra bela mrura ama proper tel bela. Engogu ayegi luror engagi damre la seng gloria bebira adril ngila dongul mo mung lamos el ngania wal gora water sample self mol talul goria. Indi akbadil la seng gora limited mo kada continue lut mora tele del mora ayegi del mora mora kele de morat mol ak madil la serva mo ngamo make sure nut el mga water sample ayegi guk kagrous el area.
Tira kasi lara lata malmo ya technology evolve so like to develop موت البيت التلة لا دغلك تواري سامبل راتمول so فلين دريق السبعة العذاب تلمرة جد التكنولوجي and then أسبوع المرورة سوبيلا ذا بلا
I see. Moral ba siyempre, kung nasa moral mo, ay technology di moral mo, sometimes maybe, who knows, as we go into the future, the technology get advanced, there might be an easier way to collect water sample without a research vessel or a big ROV, who knows, they might even have like a strong AI robot or something that it just got on there, collect sample by itself, who knows? But hopefully, in the future, we're gonna have a more easily access to go to the deep and collect more. We're very happy that the visiting of our research person has been helping the Coral Reef Center by allowing their staff to go on board and do some other work that we normally do. Without those boats, there's no way we can do those. So we're very happy that you touched that. Can we talk a bit more about how to collect? A bit in detail.
Yeah, so we the way that we typically collect is when we're offshore, so in deep waters, we end up putting a Niskin bottle that Meringue is referring to over the side. That allows the bottle to stay open until it gets to the depth that you want, and then you send a messenger, a weight, that basically goes down and captures the water at the depth that you want it. Then you bring that back up on the boat, and then we can, when we're offshore, then we filter it on the boat. When we're collecting near shore, we'll bring it back to Pickerick and do the filtration back at the lab. But we're able to do that on the marine law vessel because we bring all the equipment on to be able to collect offshore and do all the filtering there. So then you end up collecting the DNA on a filter paper, and you can then freeze that or put it into a buffer solution that stabilizes the DNA. And then ultimately we then extract the DNA from those samples. And then we use a sequencing technology that allows us to then read out the genetic code of those samples. And that then allows us to identify the different vertebrate species.
I mean, listening to you sort of, when we first talk about water sample collection, it sounds like, you just scoop the water and that's it. But when you go through it, just explain how to. That's also a lot of work.
Yeah,
I mean, a lot of little work, careful work, a lot of steps. Yeah.
It's also important. I'll note that what we're targeting are vertebrates, and we as humans are also vertebrates, which we can see a lot of human DNA in the samples as well. So it's important that when we do these methods, we also conduct them in a clean way so that we're not contaminating the samples, because what we ultimately want to be able to identify are the marine biodiversity and not just our own sequences. And so it's a lot of steps, but it also requires a lot of training so that the people collecting the samples and processing the samples know how to handle them so that we don't add additional DNA to the samples.
Oh, wow. A lot of little detail behind the water sample collection. Mulko ko lina say, umbral ko uidil, le kadamo ma jagi del water sample may, yal sumtanga ngara kala alamol, ete dia segi de mi. Ngaria tleta los peril mal make sure tomongil mi, leng giril guktio mesra ayele do aseng vertebrate ayel armlo iyuis. Mangiril mi, dice la ngara insera wasay. Mo misindel kladi energi da ada tel de bela nisyor. Ako nasa nga gidile mora ergidlo melbal marang direk mo kloa kladi energi da tel nisyor. Mendirek ngari tele trail sellomi temdura gil tele wasay. Mo ngada sal melasel di en era kada misindel melasel di en era gidil arm shad mo mesen. Which make me curious. When you do the filtration, you can filter by removing the human DNA and just the focus on.
Yeah, so the way we try to do that is by having it enclosed. So when we collect the water samples, we put them in sterile bags, and then those sterile bags when we transfer it to the filter, it stays all enclosed, protected from the outside environment. And so then we're capturing the majority of the DNA that's associated with the water sample and not the environment that we're collecting or filtering the sample in.
I still don't get it. I mean, when you say you're collecting water from the niche, right?
Mm-hmm.
And you filter it, and you say you keep it in the enclosed.
Yeah, in a sterile bag.
So what do you mean by contaminated by human DNA?
So as we go through those steps of filtration and then also downstream steps of extracting the DNA and doing all of that, there's the potential as you're working with
Like
them.
your sweat.
Yes.
Oh.
Or your skin cells.
Okay,
anything to be able to get into the sample.
That'll explain it. I was thinking of it in a different way, so I explained to the public in a different way.
Oh, okay.
I was saying when they filter, there's especially the niche, there's a lot of human DNA, maybe from our skin when we do the fishing, and we also fishing in the water. Sorry,
It's true that within the samples we also see a higher human signature, so more human DNA when you're closer to shore than when you're offshore.
Right.
So yes, you do see that as well.
But we can filter that out in the end when you get the data back. We see, oh, there is human in this sample, so there were humans around, and that is fine. But we are interested in the other biodiversity as well. So if there's a lot of human DNA, we just get less of the rest, basically. That's why it's.
I see. And niak sel gulo ko niya uktial sal alwid. Ikagila ko ni rasa niak umaral wai say. Hindi sel lulo ko niya sel lomi ra extra offshore. Ngaria ta treta lagi mi lelakin sel digas pengel ngal adromi yang drop primordial luar sample yang contaminatory. Maun ngai rasa agimi aigal DNA aigal armanabab e mal mechura gis mura ora enclosed mendia ka ora kals pengela adang. Ai. Saya lulo ko ringa nga ka ora mendia ko hiligam. Ending mo la say emerald wai say sen buk kalektra ni sure. Ang kla human la gagidlo o abangaraeng arwaskin gid mara mumraklom seguida tamal miralap selangal miralap ang kagida chinin mulwa selangal miralap e bolomi seguida tir otob da gid roguil el gora ADN era hukra de telamum de sengwara ADN era indim blo selo seguida en wadi bol que greis el DN en suadlo mesin el otob da roguil wadi bol que greis seguida los goya chinin. Wow, it's a lot of meticulous work, something that somebody must have a patience, must have a patience and sit down there and wow. I'm more like a field person. I had no idea what's going on with this. I love work. Anyway.
But not only patience, it's also كده like malagarigik la للأن سل دورير لا بنجل إذا إن أين ديجال إما ذا المرة إيتال البارل أو تال توتو الغريني أوار سامبل ريني يا ذي إن إيرجي ذا ذريقة السئل contaminate ريني.
Oh wow.
Yeah, مثل سقوم مغذا process sample إذا إن أين دخل سأم الغرام نين then منقصر أبدلو من then مرا إرتال sample نقرم الشن ساكلاف سرقة and then you always have to wear gloves نقرم لو كلاف سرقة قلتام من دخل سأم basically mali mirad as المرو لسل process ling dakal giram المرو angarang mangarang ling dakal some contaminated tel sample.
Oh wow. So this is like really sterile. When you try to extract DNA or filter, it sounds like everything has to be sterile,
Yes.
Yeah.
Try to reduce the amount of
Right.
of contaminated surfaces and things that are
Right.
acting with it. So yeah.
Man, that's a lot of like little. Wow.
Mal meticulous.
Yes, definitely.
And so for the overall, we've been working with COS, or COS has been assisting Coral Reef or Palawan Buenta doing all this. What sort of like a long term that we wanted to? What are the key goals of this EDA process?
Yeah. So initially, this was brought about by a need for being able to really understand what the biodiversity was within Palau National Waters. And so initially, our goal is to collect an inventory of the biodiversity that exists there. And so really, it's building this list of species that we're able to identify and having that baseline. And then from there, we can start to understand how changes that we see in the data might be reflecting the changes in the species. So species that maybe migrate and move around, or species that are being impacted by different environmental conditions. And so the goal ultimately is to be able to use this as a monitoring tool as we have this baseline understanding, and then being able to see how things change.
Nga singli da ko lin mo ko siya sel gora wal mo te kam le so ko akut mekli sel wal le la ba sa ron mal ben sel baseline mung le bela dra kam kat mekli da yidlo dor mela te idri te wabol do da te luror ya kada sai gid o gore mke ko gid gora abuil dringi yang mo ko la sai nsbel lo ga gid ya kada mo mngira sai kada kan tinur galet ya gid bisata idene asel dira subdi la sai wara kan dishan sai diriga wasay gida igal kaklos tukal badgal aklarada bangari alu bemna modya kadi ni rakburak pelan mun amla mok ne
Or la migrate.
alu bemu pnabral to mung bral sel doral mura future wasay rospete gidel lurol ratora idna lugora diriga ospedil monitor tial PNMS min dakan necessary to todo el mundo gora bras lom urak bongora mara sel dio ga gida mo nasi aku kora lagi kena kora maral saya malu bendikia lem tiga DNA ini saya kora wajib jadi so that's really good and we're happy you have sort of build like the baseline so from here on we then continue to build on that baseline and use it as to kind of monitor the changes in our PNMS into the future.
Yeah,
I bet that Jeanine work is quite not as hard as Palau because I'm sure California already has years and years of collection. Is that can you talk about a bit more about that? How this DNA was continued to build, and if there's any example of things that was in there and now it's gone, or just a bit more. I mean, I guess talk about the advantage of a long-term collection. What can it tell us?
Yeah, so I think Collin can speak to that too. But I think in California, there's definitely more knowledge about what is in those areas because there's more data to build on as well, and like different groups working on it. So there are databases to corroborate kind of like the eDNA collections. And also here, it's such a high diversity. So there's so many species here. And I think that is also really interesting why eDNA is such a great tool for these areas, because we are able to identify things that otherwise we would probably miss. So it is a bit different, the two systems, I would say. But yeah, it's definitely interesting to be able to use this method to describe these very, very like high diverse places. And then some of the species, I think what is a challenge here is that not all of the species are really in those databases that we talked about before. So we do get back genetic signals from things that are not identified yet.
Right.
I think that is something really cool, but yeah, it's challenging then to interpret.
Yeah, you're right, Jeanine. The more the ocean become diverse, I mean, that's a lot of eDNA too. So you're right. The rest species, I guess, is a bit easier because you only need to identify a few.
That's right.
Yeah.
But I think the advantage here of being able to use eDNA because it's so diverse, it would be almost impossible to be able to identify all of the organisms here. And so, when we collect a sample or multiple samples, we're able to identify hundreds of species. And that is something that otherwise would take a lot of time to be able to do. And for a lot of those, as Jeanine was saying, they could be cryptic species. So species that camouflage well or are more hidden or have a diurnal process where during the day they might be very deep, but at the night they come up to the surface.
Right.
And so all of these things allow us to be able to identify these organisms that otherwise might have characteristics that make them hard to identify. And so eDNA really is an advantage to be able to identify all of those organisms with just a few water samples.
And so far with this DNA collection, of course you've seen so of course there's definitely a difference between like the top layer and bottom layer. Have you ever got the chance to kind of look at the samples that Maring mentioned
From the deep,
from the deep?
those have not been processed yet.
Okay.
But our plan is to process them in the next phase of the project. But we have seen really interesting patterns even in the shallow samples. So, for example, we have species that we consider cosmopolitan species, where we see them in a lot of the different spaces. So whether it's from offshore or near shore, we will still identify those species. So different species of flying fish, for example, we see in both spaces. But then there's also species that are quite unique and that we only identify in one or two places. So one example that was quite interesting was Sheely's eel, which is a type of fish species that burrows into the sand. And we identified that only in Peleliu, where we took samples in a very shallow seagrass area. And so it's not that it's unique solely to that space, but it's more that because it's such a shallow area that we sample from, we're able to get species that really interact with the benthos, with the bottom. And so that starts to characterize more the different species that live in some of these different ecosystems that otherwise might be overlooked because they're burrowed into the sand.
I see. Mo ko lilo sing jagil sampol kalekta. Adi lo sil lo spell war identify gel arm de kan necessary ka demis. Lo ko ramalak. Yang ari atal arm lo ko ringi el dire lo mesay gal ini sampol hindi kal daseng gukmal di ngarames. Ngarames el dire kal temutiga sampol rilat bol mak. So wow, maral interesting ay gal. El to ra ini. Sayak lungel tagidel ngora walbeta dongispa. Hindi kita mo omi am karang nding gora wal bet lurore il subdi rasa ngara bangaral karo subdi la armak kel dubila. Wow. So just a quick plan. So what's the plan for the future?
So we will continue doing this work. We just received funding recently to be able to conduct this work for additional 3 years. And the goal for that next part is to be able to bring many more of the steps to Palau to be able to process and be able to sequence on island. And so that will allow us to more quickly turn around from collection of samples to data and insights from that. And so the idea is for there to be trainings on island, and also for that, for Pickerick Center to be more of a training hub for the region, so that it's not just Palau that's able to collect these samples, but also other islands around Micronesia. And, really at this point, we are starting to build a very valuable, important database of all of these samples and data that we've collected over time that starts to tell a story of what is happening within Palau's ocean space. And this is the longest time series that we have for the National Marine Sanctuary. So that allows us basically over the last 3.5 years to be able to see the different species that have the National Marine Sanctuary as part of their home, but also to see how those might change, in the coming years as we continue to sample them.
I see. Mulgol wasing tora malat kel wase aloral mobdul matat. Yakata mulmol mumru lagay dulur. Din ko na sorry wase ko sikro lasing. Sebedel ko amugut maklia sel ko na wabol bila urur belaw. Mumodya kal bailal do ko na oldurok legal more outside mat ora oldurok lagi del sampol ergi del mo matmu rai krele ko minit til oldurok kol mei agora plan el gira future ngi da ngal golin mo wasi ta amla al mut no agora geso do del mora vai gale der rak mo gora soril lo gi segui de gora mlas wasi gel se del ko makritial apply tel center el mos el ngaria agora abli la urol se del se dalek ta sampol edmei yagdi ngat tagi del bela agora mi agut mok mo mo dangile lo sengara momo dinga tiagut bok.
أساس الأقبلين رجينا أصاب نجال researcher نجاي أم أورير سجد ال EVNA لا بالغوك ثيرة لا process سجد الsample EVNA الجدا
Oh, ya mati ama ato ko ya mare.
Oy.
Oh, tiam ato ko ya ko mga kakakura. Adi aksal mo nga igal kora plan ra sentera ko to kora research. Tiam de un maka do mo ra programa kle announce ra sio ko ngel research. Tiam ato ko ito ba ko to please sigidel. Inga kita kolil mo ko si. Nso dal ko mo please sigid. Mo bet kita amru la uror belaw. Amru la galu reret dingatia omi. Ya o si silta men sadro ko si. Ya sebel ko ko kol ko sa igal ko neighboring countries especially Micronesia region. Mosbel gorawal hap gorawal another I guess ilbuk ma instead of drok mo hawang brigal eto drok mo bela mo kadu karbela or was tingal labor gidi ya ti amalung il plan emring. Wow, that's a very good I mean I'm kind of excited to have that here not only because it will kind of build the capacity, local capacity, but also the advantage of not sending these things, outside for. Right. That's a very good. Sounds really like a great plan into the future as we continue our work with. So far, all the data you've been collecting since 2021, can you just tell us a little bit about? Is there any? Are you beginning to see a story or a picture?
Sure, yeah. I mean, I think largely what we're identifying are a lot of the species that we expect to be in those spaces. So within the forereef, we're seeing a lot of the fish that we expect that are reef associated. But in the PNMS and offshore locations, I think that's where really interesting stories of the different species that we identify there, different tunas, etc. But that there's this connectivity between, it's not just the offshore and the nearshore, but rather there's connectivity between those spaces. So the organisms use the nearshore as nursery grounds, and also the younger fish go offshore. So we start to see the connections between those different spaces and start to really understand then the value of these spaces that are otherwise very far and you don't think about very often, but are really important for the life cycle of these species.
I see. Oh, isel otro molgo kolya sa sel otro statalek taigal inel merelang yung korawa blabul blok klasi ngal me eda matmol adire kol korawal kau kwak aling sele ora connectivity tit blok klasi bebira ngais rangi ngarat mol adire kol me uspera me eda korawal loral mo klo ku mutod morawal mo waseng ogay kayo de talbal galekte en wa blabol se de sa sel value maluben sel korawalul tutlel yal offshore ngula kalmera nisyon mambrula say hindi ka necessary sa kaduk mal kora di manage sa tel offshore ma ngired kya korawal gired kora bet shureng hindi ka matmala say gidel el tokora rolela ngidel ma mong malungi le am sira dulu temeral mong yung mga mo toka ko la kakros betul la arma sa el mesel ano kanya kasama mo. Well, Collin, Jeanine, thank you so much for all this work you're doing with us in at the center, especially for Palau. It's important work, and I think we should continue to build on the baseline you create so far. So in the future, the young generations, when they grow up, they can continue to take over this work and continue to manage a PNMS, and I'm sure they'll be happy and saying, man, our four brothers and sisters did a really great job. So now we can just sit in the lab and continue to do this. Anyway, we have come to the end of the show. So if you want to say anything, before we conclude, I just wanted to say, not only you, all the partners involved in this work, we really appreciate their time and effort, especially the Marine Law, you guys, people researchers, and so I just want to emphasize the fact that this collaboration and people's efforts and time would make this work possible. So I really appreciate everyone's.
It would not be possible without Ministry of Justice and being able to use the boats and work with the crew at marine law to be able to collect these
All right.
samples. So
Yeah.
that is part of the initial part of collection, and then the Pickrick researchers are doing the work that follows after the collections. But that collaboration has been really important to being able to make this successful.
And since we don't have a bigger research vessel about yet, I'm sure they're happy to continue to assist us in that regard. So, yeah, we really appreciate their effort. Jeanine, did you want to say anything before we close?
Well, I just want to say thank you for having me. I've never been in Palau before. I saw some of the data, and it's very fascinating because it's so diverse. So I'm an avid diver. I really like diverse tropical places to dive. But yeah, it's been great to look into the data, but now actually being here and like learning from the people at BUKURROU and see the environment. I look forward to it. So yeah, thank you for having me.
I'm sure as I said, then both of you will cross paths in the future for sure. And Marek, did you want to have a final comment before we close?
No, maldiso kalorang sulit our listeners, sumatri del collaborators, sirgi del luuling sa edel makatmok latialorior. Evisa kalo ay silmot siyanin na makalin, manggel chief researcher, gidara ikalaw oto, amora Monday tamra lalmar aqua malmobor dra okeanos emro ngesel elmer vela wal malmal mertelurier al galekta sample DNA.
Oh wow. Oh, material teru lang ang tamra mandi.
Oo, tamra
Kalkalin matinin manggel lolgoria igalaw oto. Amomra mandel mong lang ari segi del dial okeanos setto dra aqua be kupra elmer vela.
Paise
Selme ngmel ku cover tel de vela.
oo, nutamosto breger ogul station ergidra DNA el miliwekler vela we galekta water sample el modo alela ay gal deiral already ngaringiri.
Yeah, man, I'm so happy that this kind of work will continue. I just learned now that you guys are going to come and you're going to board. I didn't know all this, so I feel really happy for all this arrangement that you guys been working on to making sure that when the ship is here you can continue to collect. Wow, I'm really happy. I'm really happy about the kind of things we've been doing in our Palau National Marine Sanctuary. Okay, always saying gora kadmara ulo gora siorgit mga kakmal mo na sulim na smiltunin ngay segit seldial mo ngarse may akmadingel wasya kukleme rula gelureri rebol meret ang sedel mutumeti away sa simong ngaraku ko gora amloke karegal lureri sel meseldal bo kuklutul maral miliwek ngay kita amregal mo wasay tomo maral miliwek mutumura yegi del basyo البلال نيل تروى مريغ لسي اجا جدا يجي دولا وار سامبل ني مثل ده المره يجي يدرس بس يعمل وار سامبل من سيد رو كومس لسي مال بولومي ايغال وار سامبل ندير غواي سي جل ني دير كيرا يجال من غير روك نامو ديبوس مال بنغو بيس نا مسويس مال ولا رو تريغال رو موكل يا كده مال ملاي تراتا مال ايامار مال دي مريغا سولين مسميل توني لورين ستيال سيرغي رابريس الايس a siorgida di mettiamo musim mesel gukmel belsel o mal sulak
arangalage balwa magabe arangalage magale